Um, methyl groups, unlike acetyl groups, stimulate the DNA to fold into heterochromatin. And additionally, the addition of phosphate groups are kind of act like the acetylation. The histone code hypothesis states that specific combinations of modifications and the order in which they occur help to determine the chromatin structure and thus influence gene expression and transcription. So to go over this briefly, DNA acetylation and phosphorylation promote the euchromatin shape and thus the form for active gene expression, because it's it's more loosely bound, so it's more accessible. And DNA methylation promotes the coiling of chromatin into heterochromatin, and thus brings about a form known for having inactive genes. So Maggie, is heterochromatin tightly or loosely wound DNA?
Tightly.
And Jack, is acetylation promoting gene expression or inhibiting it?
It promotes it because it loosens the chromatin.
Yes, it does. Okay. So some enzymes methylate the tails of histone proteins. Others have the capability to methylate bases within the actual DNA. And in humans, the Barr body, which is our inactivated X chromosome in females, tend to have higher methylation in regions of operating transcription. So essentially, because it's not used in the cell, because it's the inactive X gene, we have it wound into heterochromatin because we don't need it to be loosened and taking up space in the cell. So it's more heavily methylated. Just like on that note, genes in cells where they're not expressed or conveyed have the propensity to be more methylated than in cells which they are expressed. So if a cell isn't using a gene, it's going to be more methylated, and in a different cell where it needs that gene to be expressed, it's not going to be as methylated, it's probably going to be acetylated. And in some studies, the lack of methylation enzymes have led to aberrant embryonic development. Essentially, there's problems with the organism developing forward. So DNA that's methylated appears to pass on the methylated quality through sequential cell division. So if in a cell a gene is methylated and then that cell divides in the two daughter cells, the gene is going to continue to pass on the methylation, so both of those genes are going to be methylated. Okay? Any questions so far? Great. As I just said, the DNA sites that are already methylated methylate the correct daughter strand in DNA replication and in sequential cell divisions. The cells of specialized tissues have sort of chemical records for methylation, which essentially says what I just said, that they continue to methylate the cells over a series of divisions. Genomic imprinting states that, methylation permanently regulates the expression of either the maternal or paternal allele of particular genes during embryo growth. Does anybody have any questions on that?
I don't get it.
Okay, so genomic imprinting it's kind of like a gene is expressed either from your mom or your dad, and depending on, and let's say it's expressed from your mom, then it's going to silence that paternal gene by methylating it, and then over those cell divisions, you're going to continue to express the maternal gene because they're going to continually suppress the paternal gene with methylation.
That makes sense.
Okay, great. Epigenetic inheritance, this is a little review because I know we learned it last semester. So although the methylation of genes does not affect the sequence of the actual DNA, doesn't change the nucleotides, it is continually passed on in future cell generations, which kind of seems counterintuitive to what we already know, where everything we have is expressed because of our coding of our DNA. But as we've talked about before, epigenetic inheritance talks about the influence of outside, such as environmental factors that affect how we express our DNA. So epigenetic inheritance is the inheritance of traits transmitted by mechanisms not directly involving the nucleotide sequence, and modifications to the chromatin can be reversed such as acetylating, deacetylating, methylating, demethylating. Chromatin modification systems can interact with each other to work together. Experiments propose that particular proteins first bind to methylated DNA, and then they call up histone deacetylation enzymes to duly repress transcription. So epigenetic inheritance can explain the differences between identical twins, where their DNA sequences are completely identical, but they have differing phenotypical features, or one has a disorder and the other one doesn't, such as Alzheimer's or something like that. Continuing on, we're going to talk about transcription initiation now. So we've learned so far of the modifications of the to the chromatin and how that affects transcription, such as the modifications to the histones and DNA. But now we're going to learn about how the DNA, how the cell suppresses transcription once this DNA is already in a euchromatin or a good form for transcription to take place. So let's go over the eukaryotic gene organization. So there's the transcription initiation complex, which is a cluster of proteins that assemble on the promoter sequence at the upstream end of the gene. And then after the complex is in place, RNA polymerase II binds and then transcribes the gene. The RNA is processed adding a 5' cap and a poly A tail, and then it splices out the intron, so it's left with just the exons. A review of the 5' cap consists of modified forms of guanine added to the 5' end of the messenger RNA and is synthesized before the poly A tail, and the poly A tail adds about 50 to 250 adenine nucleotides to the 3' end of the messenger RNA. The cap and tail serve to facilitate the export of mature messenger RNA from the nucleus, so it can travel from the nucleus to the cytoplasm, protest mRNA from degradation by hydrolytic enzymes so it doesn't break down in the cytoplasm, and help ribosomes attach the 5' end of the messenger RNA once it is already in the cytoplasm. We have what we call control elements, which are segments of non-coiling DNA that serve as binding sites for proteins called transcription factors. These control elements and the transcription factors bind and are essential to specific gene expression regulation in the varying cell types. So they're important in determining the difference between a muscle cell and a nerve cell and those different functions that their role is to serve in the organism. The role of the transcription factors. There are what we call general transcription factors, which are vital for transcription in all protein coding genes. And as we've learned, the TATA box within the promoter is an example of where a transcription factor binds by itself to a DNA sequence, so it's not binding with other things that bind to the sequence. It itself is binding to the TATA box. Most transcription factors bind to proteins, RNA polymerase II, or to one another. Does anybody have any questions so far?